You know that feeling when you're just surrounded by information, maybe even a little overwhelmed,
Yeah.
but you still feel like you're missing the big picture?
Oh, absolutely. Especially with something like genetics.
Exactly. It shapes so much of who we are, how we live, even how whole societies work. It's profound stuff.
It's a vast landscape for sure. Lots of data, often quite intricate.
Well, today, our mission is to take a deep dive into the sources you've pulled together. We want to help you, our listener, quickly grasp some really fascinating insights.
Yeah, we're going to unpack everything from like the complex science behind family trees.
All the way to those tricky ethical questions that pop up with things like genetic screening. It's quite the journey.
It really is. And it's remarkable how these concepts, which can seem abstract, actually play out in real lives, across different cultures, through history, even with royalty.
They really do. And we've got a great set of sources for this. We can explore core ideas, like what inbreeding really means beyond the common perception.
Right. And how we measure genetic similarity, how genes impact survival in surprising ways.
We'll also dig into some deeper stuff like genetic load and gene imprinting. Sounds complicated, but we'll break it down.
And definitely look at the tools we have now, like genetic screening and counseling. Powerful stuff, but raises big questions for the future.
Okay, let's jump right in then. To really understand our genetic blueprint, maybe we should start with one of the oldest and maybe most misunderstood concepts,
Oh,
inbreeding.
good starting point.
It turns out it's way more nuanced and honestly more widespread than I think most people realize.
It genuinely is. I mean, at its simplest, inbreeding just means mating between close relatives. Pretty straightforward definition.
Right. But the consequences genetically,
Yeah,
that's where it gets interesting, like a double-edged sword, you said.
exactly. It really can be. When close relatives have kids, there's a much higher chance those children inherit two identical copies of a specific gene. We call that homozygosity.
Homozygosity. Got it.
Now, the risk comes when both those copies happen to be uh, deleterious or harmful versions. That usually leads to reduced fitness, basically, a lower chance of thriving or reproducing.
Okay, that makes sense. That's the immediate downside people think of.
Huh.
But is it always bad? Are there like surprising long-term effects that challenge that?
Ah, this is where it gets really fascinating. Over very, very long periods, we're talking millennia, natural selection can actually step in.
Oh, so.
It can sort of purge or remove those consistently harmful gene versions from the population's gene pool.
Wow, so it can like clean house genetically. That's a wild thought.
Exactly. And potentially, this leaves behind advantageous genes that are homozygous. So while the short-term effects can be pretty bad, in the super long run, it's not always a purely negative thing.
That's powerful. Time isn't just a healer, it's like a genetic sculptor. So how do scientists actually measure this genetic closeness and the potential impact? Is there a number?
There is. We use something called the relationship coefficient, usually just called R. Think of it as a math formula to see how much genetic material two people share because of common ancestors.
Okay.
It tracks the probability, essentially, that they both inherited the exact same gene copy from someone way back.
So give us some examples. How close are we talking R value-wise?
Well, okay, first cousins.
Hmm.
They typically share about 12.5% genetic similarity based on this, so R is 0.125.
12.5%. Okay.
Yeah.
What about, say, an uncle and niece?
That jumps up to 25%, or R equals 0.25.
And siblings. I remember reading this often surprises people.
It does. Siblings share, on average, 50% genetic similarity. R equals 0.5.
50%. Wow. Okay, so how does that similarity number R translate into the actual risk for any children they might have?
Great question. That brings us to the inbreeding coefficient, or F. It's pretty simple. F is just R divided by 2.
Oh, okay.
And what F tells you is the proportion of genes in the offspring that are likely to become homozygous because of that shared ancestry. It directly quantifies the risk.
So F is directly tied to the chance of getting two identical copies of the gene.
Precisely. So if siblings were to have offspring, which is thankfully rare and often illegal, their F value would be 25%. That means a 25% chance for any given gene to be homozygous due to that shared background.
And for first cousins?
It's about 6.25%. For an uncle and niece, 12.5%. These numbers just highlight that increased chance of inheriting two copies of the same gene version could be good, could be bad.
Right. So given these risks, why are consanguineous marriages between blood relatives, still so common? The sources say like over 20% of the world's population. That's huge.
It really is a staggering number. And there are several powerful reasons behind it. Often it comes down to social stability, a feeling of higher compatibility within established cultural norms.
Less uncertainty, maybe.
Exactly that. It's often seen as better for a woman's status in some cultures. There's built-in familiarity and easier transition into the new family unit.
Fewer hidden surprises, perhaps.
Could be. Less worry about hidden health or financial issues sometimes. Plus, the whole process of premarital negotiations can just be simpler and less costly.
Makes practical sense in some contexts.
And sometimes it's just about small population size or limited choices. Think of isolated communities where people have traditionally married within their group for generations.
Like the Ashkenazi Jewish communities you mentioned earlier.
A classic example, yes. And historically, we can't ignore the blue blood royal marriages. The aim there was crystal clear. Preserve what they saw as pure bloodlines, and critically, secure the succession of power and titles.
Keep it all in the family, literally. In history, it gives us some pretty stark examples of how that played out, doesn't it?
Oh, absolutely. Take Charles Darwin, the father of evolution himself. He married his first cousin, Emma Wedgwood.
Right?
They had 10 children, but only 7 lived past age 10, and only 4 of those were fertile themselves.
That's pretty telling. What's the immediate lesson there about inbreeding's impact?
It just clearly shows that immediate short-term risk. When you introduce inbreeding, those harmful recessive traits have a much higher chance of showing up right away.
Okay. And then there's Queen Victoria.
Yes, a carrier of the gene for X-linked hemophilia, the bleeding disorder.
Right.
Because of generations of intense intermarriage among European royals, something like 20 members of various royal families ended up suffering from hemophilia, all traced back to her single gene copy. It's dramatic.
Wow. And maybe the most extreme case study is the Spanish Habsburgs.
Oh, definitely. King Charles II. The multi-generational inbreeding in that line was so intense, it led to severe health problems for him. He was famously described as having rotten organs and no blood.
Yikes. They call him Charles the Bewitched.
They did. It just painfully underscores how those harmful, deleterious traits can pile up over generations of sustained inbreeding, creating a really heavy genetic burden.
Okay, that paints a pretty grim picture of the risks. But this leads us to that paradox you mentioned. If it's so bad, why do some populations seem to do okay or even thrive with really high rates of it?
This is the inbreeding paradox, and it's fascinating. Look at Saudi Arabia's Dam province. Over 52% of marriages there were consanguineous.
More than half. You'd expect major problems.
It would. But surprisingly, there's relatively little evidence of widespread reproductive wastage, like high rates of miscarriage or severe genetic disorders being rampant.
So what's the explanation?
The leading theory points to the very long history of the practice in that region.
So time again. Allowing natural selection to do its thing.
It seems so. We're talking millennia of this practice, potentially allowing natural selection to gradually weed out the really harmful recessive gene versions over many, many generations. The population has, in a genetic sense, perhaps adapted.
And we see similar patterns in India.
Yes, the case studies are quite telling. For instance, the Ansari Muslims of Balpur, Bihar. Reports suggest they started practicing cousin marriage relatively recently, maybe 150, 200 years ago after converting.
And the effect.
Sources suggest they are currently showing signs of active inbreeding depression. Things like higher infant mortality, maybe lower average IQ scores reported in some studies. Because it's relatively new for them, the negative effects haven't been purged yet.
Whereas other groups in India, like certain Brahmin communities in Tamil Nadu and Maharashtra, have practiced it for a very long time.
Exactly. And in those groups with a long history, you don't see the same level of observed reproductive problems. So the startling insight is really about time. When did the practice start? Millennia can fundamentally change how a population responds. Evolution in action.
That's incredible. Okay, let's shift gears a bit. From these historical patterns to how genetics is shaping our future now, especially through tools like genetic screening and counseling. First off, what exactly is genetic screening?
Fundamentally, it's about testing people for specific variations in their DNA, variations that might increase their risk of developing a disease or maybe passing one on to their kids.
So finding hidden risks coded in our genes.
That's the idea. And there are 2 main types. First is carrier screening.
Carrier screening, what does that find?
It identifies people who carry just one copy of a recessive gene variant that could cause a disease. Think sickle cell or that X-linked hemophilia we talked about.
So the carriers themselves, they're usually healthy?
Typically, yes, because they usually have another normal working copy of the gene that does the job. But, and this is key, They can pass that potentially harmful recessive copy to their children.
So it's important for family planning.
Hugely important. If both parents happen to be carriers for the same recessive condition, their child has a 1 in 4 or 25% chance of inheriting both harmful copies and actually having the condition.
And for those X-linked ones like hemophilia, it's different for boys, right?
It is, because males only have one X chromosome. If they inherit a harmful gene on that X, there's no second X chromosome with a backup copy. So the condition shows up. Knowing your carrier status gives you options, information. It's empowering.
Definitely. Okay, what's the second main type of screening?
That's predictive testing. This looks for gene variations linked to an increased risk of developing disorders, often later in life.
Like what kind of disorders?
Things like certain autoimmune conditions, some forms of cancer, Parkinson's disease, Huntington's disease. But it's crucial to remember. It predicts predisposition, a higher likelihood, not certainty. Many factors usually play a role.
Got it. A risk factor, not a diagnosis in itself. And when do these screenings typically happen?
At different life stages, there's newborn screening.
The heel prick test most parents know.
Exactly, done right after birth. It screens for conditions where catching it early makes a massive difference, like PKU phenylketonuria. Just changing the baby's diet can prevent severe intellectual disability. It's life-changing.
Amazing. What other times?
Then there's prenatal testing, done during pregnancy. This screens for more severe genetic disorders in the fetus.
Helping parents prepare or make decisions.
Yes, it provides information. It might help parents prepare for a child with special needs, or sometimes leads to difficult decisions about continuing the pregnancy. Methods like amniocentesis or CVS, chorionic villus sampling are used.
And the last type.
Symptomatic testing. This is done when someone actually starts showing symptoms that suggest a genetic disorder, like spinal muscular atrophy or Huntington's. It helps doctors confirm the diagnosis.
So you get these results. Maybe a carrier status or predictive risk. What happens next? Just getting the data isn't the end, right?
Absolutely not. And this is where genetic counseling is so important. It's way more than just handing over a lab report.
It's about understanding it.
Yes, it's about communicating really complex information in a way that makes sense, that's meaningful for the individual or family, and empowering them to make informed decisions based on it.
So translation and support. What does a counselor actually do?
Well, typically it involves 3 main steps. First, they gather and interpret your family's medical and genetic history. Then they do a thorough risk assessment using that history and any test results.
Okay.
And third, and maybe most critically, they communicate all this information effectively. They help you understand what it means for you, for your health, for your family, and support you in the decision-making process.
The benefits sound huge.
Yeah.
Empowerment, better medical care.
Definitely. It supports reproductive autonomy choices about having kids, and it can really improve emotional and financial well-being by helping people prepare or make choices to avoid certain genetic outcomes.
It sounds incredibly valuable, but I can't help thinking it also feels like walking into an ethical minefield. What are some of the potential downsides or challenges here?
Oh, there are definitely complexities, big ones. One issue that can pop up is mistaken paternity.
How does that cause problems?
Well, genetic counseling relies heavily on having an accurate family history. But if that history is wrong, maybe because of different cultural ideas about fatherhood, like the Yap tribe example, or just undisclosed family secrets,
Oh,
it can lead to totally wrong risk assessments and test interpretations, and potentially some very awkward, very difficult conversations.
I can only imagine. What else?
Another major challenge is handling information about diseases that show up much later in life.
Like Huntington's again. Symptoms might not appear for decades.
Exactly. Imagine being told when you're, say, 20, that you carry a gene, meaning you'll almost certainly develop a devastating disease later on. That knowledge creates a massive, lifelong psychological weight. How do we ethically handle that kind of information, especially for kids or teenagers who can't really consent to knowing?
That's a really tough question. And then there's the bigger picture concern, right? Discrimination. As we get better at reading genetic code, could we see genetic discrimination?
It's a very serious worry. Could health insurance companies start charging people more based on their genes? Could it affect getting a job, even finding a partner, if people start sharing genetic profiles?
Wow, that sounds like something out of science fiction, but maybe not so far off.
It underlines why confidentiality is absolutely critical. Protecting genetic data is paramount to prevent what some people call genetic humiliation, or just blatant discrimination based on DNA.
So a real tension between the benefits and these potential harms. But let's also look at the positive impact. These programs are making a difference, right?
Absolutely, undeniably. In India, for example, screening Sahara tribes helped identify immune system genes linked to TB susceptibility or resistance. That's vital public health knowledge for that community.
Understanding population risk factors. Makes sense.
Precisely. And look at that Gujarat Red Cross program from 2016. They screened 300,000 pregnant women. The reports say it prevented 186 births of children who would have had severe thalassemia, a serious blood disorder. Gujarat's become a model for dealing with these conditions.
That's a very concrete, positive outcome.
Extremely tangible. And think about India's National Sickle Cell Elimination Program aiming to screen 70 million people by 2047. It's incredibly ambitious, aiming to essentially eliminate the disease through screening and counseling.
Wow, 70 million.
And one more example. The Ashkenazi Jewish community, particularly in the US, they have higher frequencies of certain recessive disorders like Tay-Sachs, partly due to historical marriage patterns. Widespread carrier screening programs have become common, helping people make informed choices and dramatically reducing the number of babies born with these conditions.
So, when done thoughtfully, with counseling and support, these programs clearly change lives and improve public health. The potential is enormous.
Without a doubt.
Okay, let's dig even deeper now into some of the, let's say, hidden costs and just plain weird things coded in our genes, starting with something that sounds pretty dark. Lethal genes.
Yeah, the name kind of gives it away, doesn't it?
Yeah.
Genes that are literally like ticking time bombs.
Basically, yeah. These are genes carrying instructions so flawed that they cause the death of the individual who inherits them, often before they can even reproduce.
How did scientists first figure these out?
A classic early example is from Lucien Queneau back in the early 1900s. He was studying coat color in mice, agouti mice. He noticed a specific genetic combination of homozygous mutant was just missing from the expected offspring ratios. It never appeared.
Right. He deduced it must be lethal. The absence of those mice told the story. Their genetic makeup prevented them from surviving.
That's clever detective work. Are there different kinds of these lethal genes?
Yep, a few categories. You've got dominant lethal genes. Just one copy is enough to be fatal, though sometimes, like with Huntington's, the lethal effect happens later in life, after reproductive age.
Okay, what else?
Then recessive lethal genes. Here, you need 2 copies to cause death. Think of severe forms of sickle cell anemia or cystic fibrosis. Inheriting 1 copy usually makes you a carrier, but 2 copies cause the lethal condition.
Got it. 1 copy. Okay, 2 copies fatal.
Exactly. And then there are conditional lethal genes. These are only deadly under specific environmental conditions. PKU is like this. It's lethal if you eat food with phenylalanine, but manageable if you avoid it. Or G6PD deficiency eating fava beans can trigger a severe, potentially fatal reaction called favism in people with this gene.
So the environment pulls the trigger, basically.
Precisely. Now, not all harmful genes are instantly lethal. The sources also talk about semi-lethal genes. These cause death in most, but maybe not all, individuals with the problematic genes, say less than 90% mortality, but still a very high risk.
Like hemophilia A?
Yes, that X-linked clotting disorder is a good example. The severe lack of clotting factor means a high risk of bleeding to death, even from minor injuries. Not everyone with the gene dies from it, but the risk is way higher.
Okay, so it's clearly a spectrum lethal, semi-lethal. How do we classify genes more broadly by their impact on just, well, surviving and thriving?
It really is a spectrum. At one end, you have vital genes. These are the absolute must-haves, the foundation. They handle routine, essential cell functions, sometimes called housekeeping genes, stuff we literally can't live without.
Like what?
Think of the TP53 gene, a major tumor suppressor. Or the SMN1 gene, essential for motor neurons mutations, cause spinal muscular atrophy, SMA. Defects in vital genes drastically reduce survival chances.
Okay, then what's next down the scale?
Then you have subvital genes. These aren't lethal, but they're like inferior quality. They reduce overall viability or efficiency, making individuals more likely to get sick or die younger.
Examples?
The FTO gene, strongly linked to obesity risk, is one, or certain variants of the APOA4 gene associated with higher risks for Alzheimer's or heart disease. They increase risk, they don't guarantee a bad outcome, but they lower overall robustness.
And the flip side, are there genes that actually give an advantage?
Yes, those are super vital genes. These are superior quality genes providing a clear survival edge. Think disease resistance. That CCR5 delta 32 variant we mentioned gives resistance to HIV. or certain HLA alleles like DRB116 offering resistance to TB.
Which again brings up those ethical questions about identifying or even selecting for these superior genes down the line. Fascinating, but maybe scary.
It definitely opens up that conversation.
Okay, sticking to the concepts, let's talk about genetic load. Sounds heavy.
It kind of is, yeah. Think of it as the collective genetic baggage a whole population carries around. It's the overall reduction in fitness survival, reproduction across the population because of all the harmful genes and combinations floating around.
So it's like a measure of the genetic imperfections in the group.
Sort of, yeah. It represents the proportion of individuals who might die early or fail to reproduce due to their genes, and its significance is, well, profound and maybe a bit counterintuitive. It acts like a thinner for the population.
A thinner. How is that helpful?
By removing individuals who are less genetically fit, it helps keep the population adapted to its environment and within its resource limits. Paradoxically, over time, this can actually increase the average fitness of the survivors.
Wow. Okay, so it's like a necessary evil for long-term population health. Are there different ways this load builds up?
Yes, several sources contribute. There's mutational load. This just happens naturally because new mutations pop up spontaneously all the time. Random DNA copying errors. About a third of hemophilia cases, for example, are new mutations, not inherited.
Just bad luck in the genetic lottery.
Pretty much. Then there's inbreeding load or inbreeding depression, which we've discussed. It comes from more mating between relatives, which makes those hidden harmful recessive genes appear more often, like the Ansari Muslims example again.
Got it. What else?
There's migrational load. This occurs when individuals or groups with genes adapted to one environment move to a different, unsuitable one.
Oh, like the example of fair-skinned people moving to really sunny places.
Exactly. Or people adapted to low altitudes, moving high up into the mountains. Their bodies aren't suited to the new conditions, creating a genetic burden or load. A pregnant woman moving to high altitude might face complications like stillbirth because her physiology isn't adapted.
The mismatch causes problems. Makes sense. Okay, that covers a lot about the burdens in our genes. Now, for something really different, maybe surprising, gene imprinting. We usually learn we get genes equally, right? One copy from mom, one from dad, both working.
That's the standard rule, yeah. Called the principle of equivalence. Normally, both copies of a gene you inherit are active.
But imprinting breaks that rule.
It's a fascinating exception. With imprinted genes, only one copy, either the one from your mother or the one from your father, is actually switched on and active. The other copy is silenced, turned off permanently in the relevant body tissues.
Turned off completely for that person's whole life. That's wild.
It is. It's not super common. It only applies to a small fraction of our genes, but they're important ones. We see it in mammals like us and also in flowering plants.
Can you give a clear example?
Sure. A key one is the IGF2 gene. It plays a role in fetal growth. In mammals, typically only the copy you inherit from your father is active. The copy from your mother is silent.
Only the dad's copy works for that gene. Why? Why silence one perfectly good copy? Seems inefficient.
Well, it does seem odd at first. There are a couple of main ideas or hypotheses about why it evolved. One is called the conflict hypothesis, sometimes called the parental tug-of-war.
A tug of war between the parents' genes. Seriously.
In an evolutionary sense, yes. The idea is there's a conflict of interest. Paternal genes from the father benefit from maximizing the growth of the current fetus, getting as many resources from the mother as possible. So paternal genes promoting growth, like IGF2, tend to stay active.
Okay.
But maternal genes from the mother have an interest in conserving the mother's resources, maybe for future pregnancies with potentially different fathers. So maternal genes that might promote too much growth could be silenced.
Whoa. A biological battle of the sexes playing out at the level of our genes. That's amazing. Evolution isn't always cooperative then.
Not always, no. It can be a conflict over resources. The other main theory is quite different, the prevention of parthenogenesis's hypothesis.
Preventing virgin birth.
Essentially, yes. This idea suggests imprinting evolved to make sure you absolutely need genetic input from both a male and a female parent to create a viable offspring.
How would imprinting ensure that?
By silencing different sets of essential genes on the maternal chromosomes versus the paternal chromosomes. If you only had maternal chromosomes, certain essential paternal-only active genes would be missing. If you only had paternal, key, maternal-only active genes would be missing. Development couldn't proceed normally.
So neither an egg nor a sperm could develop fully on its own. You need both active sets.
Exactly. It guarantees the need for sexual reproduction. A pretty neat evolutionary trick.
Okay. So imprinting is this delicate balance. What happens if it goes wrong?
Right. Things can go awry. A rare error called uniparental dysethemy, or UPD, can cause problems.
Uniparental, from one parent.
Correct. Normally, you get one of each chromosome from mom, one from dad. With UPD, you mistakenly inherit both copies of a particular chromosome from just one parent.
Does that always cause issues?
Not necessarily. It only really becomes a problem if the chromosome involved happens to carry imprinted genes.
Ah, because then you mess up the balance. You might get two active copies when you need one, or two silenced copies.
Precisely. You disrupt that critical dosage, leading to specific developmental syndromes. A classic pair involves chromosome 15. One is Prader-Willi syndrome.
Okay, what happens there?
This usually results from either losing the normally active paternal region on chromosome 15 or inheriting both copies of chromosome 15 from the mother maternal UPD. You're missing the active paternal contribution.
And the symptoms.
Things like excessive sleeping early on, later an insatiable appetite leading to obesity, weak muscle tone, often infertility. Little memory hint: P for paternal problem, P for Prader-Willi.
Got it. And the flip side involves the maternal copy.
Yes, that leads to Angelman syndrome. This happens if you lose the normally active maternal region on chromosome 15, or if you inherit both copies from the father paternal UPD, missing the active maternal input this time.
And the symptoms are different.
Quite different, yes. Often includes epilepsy, tremors, developmental delays, and a characteristic happy demeanor, frequent smiling or laughing. Memory hint: Angel for the maternal dilution aspect.
Wow. So these two syndromes dramatically show how critical that parent-specific gene activity is. It's like this incredibly intricate choreography inside our cells, and one tiny misstep has huge consequences.
They absolutely do. A powerful illustration of the delicate balance required for normal development.
Okay, so wrapping this incredible deep dive up, what does all this mean for you, our listener? We've gone from hidden layers in your family tree, maybe, to thinking about health, the future of medicine.
Yeah, these insights into things like inbreeding, screening, genetic load, imprinting, they really reveal the amazing complex blueprint that makes us who we are.
It's a powerful reminder, isn't it? Our genes carry this complex tapestry: vulnerabilities, strengths, advantages, all shaped by thousands of years of evolution.
But now, also increasingly shaped by our own scientific discoveries and the choices we make as a society.
Absolutely. And while a lot of our genetic story is written down in our DNA, how we understand it, how we respond to it, that part is definitely still evolving.
Which brings us to maybe the big closing thought. As we gather more and more genetic data, and it seems inevitable, it really raises a crucial question for you to think about.
How are we as a society going to balance the incredible benefits of all this genetic insight, which we've seen can be immense, with that absolutely critical need for privacy, for fairness, and for preventing discrimination based on DNA in the future?
That's the tightrope we have to walk.
It really is. Definitely something to mull over until our next deep dive.